Thanks for joining me. I'm going to talk about some work that we've been doing applying eDNA methods to assessment and management of estuarine systems. My name is Allison Watts. I'm at the University of New Hampshire. And I'm in the Department of Civil and Environmental Engineering, which means that I'm not a biologist, but I get to play with them occasionally. And here's me with Bree Yednock in South Salute, Oregon, pretending to be a biologist. The work that I'm going to talk about is something we've been working on for the last couple of years with the National Estuarine Research Reserve System. So we've been working with 6 different reserves across the country, and these are the people who are at those reserves who have been our partners in the project. So the staff of the reserves do the sampling, collect the samples, give us advice, give us feedback, and are really the backbone of this project. Once the samples come to the University of New Hampshire, we analyze them, we take those results, we go back to the resource managers that we're working with, and we try and understand how they can use those results and how they're relevant to their management needs. So the project that we're talking about, something, as I said, I've been working on for a few years. It's a collaborative project where we work very closely with researchers and end users together. And we have several goals here, but the one I'm going to talk about is we wanted to try and develop some eDNA sampling collection and analysis protocols that were relevant to the National Estuarine Research Reserve System for using eDNA in estuarine monitoring. And there are other aspects of this project I'm going to talk today pretty much about our recommendations and some of our results related to fish. If you're interested, Devin Thomas has a poster on biological monitoring distinct habitats using eDNA. That's a poster somehow. I'm sure you can look it up and find it. So today I'm going to talk about fish and recommendations that we came up with for fish. So the NERS, the National Estuarine Research Reserve System, all do standardized monitoring. They go out every month. They have a set of sites that they sample, so they are out there taking water samples every month. What we wanted to do was understand what would be a workable, so doable, not too much insane amount of work, addition to their existing sampling program that would allow them to incorporate eDNA into their sampling protocol. And we looked at a lot of different methods. There's tons of great work and information out there. We didn't invent anything. So we took a lot of different methods. We looked at what we thought would work for our particular sites. And we wound up recommending triplicate 1-liter samples at each of the sampling locations. And there are different recommendations in the literature. We found that for single samples, so this is sample locations in Great Bay, New Hampshire, and number of fish species detected. So for the number of locations, and this is one sampling run in June, for the number of locations sampled, the number of species go up. If you take triplicate samples, 3 1-liter samples at each location, you get significantly more species at each site. And when you're out there in a boat, going to a new location is a ton of work. Just grabbing another 2 liters of sample is not as much work. You still have to filter them and analyze them, but it's not as much work. So sort of bang for the buck, we felt that triplicate samples was a reasonable compromise. We take these back and we filter them in the lab. Many people prefer to field filter. Again, if you're in a boat and the tide's going down, we found that people just didn't have time to do the filtering in the boat. So it just is more practical in this situation to take it back to the lab and filter it. And this is Chris Peter at Great Bay Near, and you can tell this is actually 2020 by the mask that he's wearing. We also find that estuarine samples tend to be very turbid. And if we're thinking about a standardized monitoring program that estuaries can use and compare results, we felt that we needed to have a filter size that could pass a liter without too much effort. So we went with 1.5 micron filters. It's a little bit larger than many people use. But if we went with a smaller filter, we found that we just couldn't get a liter through them in most circumstances. So again, that's a time and an effort compromise that we decided to make. And this is a filter, and that's what an average filter in Great Bay looks like in August. Some of them look worse, sometimes even with 1.5. It takes a couple of filters to get them through. For fish, we're using a 12S primer, the MyFish primer that many people use. We use Kyogen extraction kits, so we're really trying to do a standardized program that many people will be able to replicate. So using those methods, some sort of general results that we came up with. This is in Great Bay, New Hampshire again. One of the things we're interested in is how many samples do you have to take in order to characterize the fish species that are present? So these were samples that were collected over the course of the summer. It's about 130 samples. And what we found is that even with that many samples, given that they're taken at different locations and different times, we continue to see more species as we collect more samples. So this is the average, sort of the mean number of species collected per sample. Obviously, there's a lot of variation. Some samples you might get none. Some samples we see up to like 7 or 8 species in a sample. So it's very variable. So we took that information and we tried to look at that a little bit more closely and understand what species we're detecting. These are the fish species that we expect to see present in Great Bay. So in blue, this blue circle are species that are likely to be present, and the yellow species are species that we detected with eDNA in 2019. And you can see that there's a lot of overlap. All of the common species were detected. There are some species that we might expect to see that we didn't detect, and maybe they just weren't there at that time. Some of this is a technical issue. We have difficulty resolving flounder with this particular primer. So we do detect flounder, but we can't resolve it to the species. So that is more of a technical issue than a sampling issue. The other thing we found is that we see a whole lot of species that are probably not actually present in the Great Bay Estuary. So these are freshwater species, and they are probably related to DNA that's coming into the estuary from the freshwater river. So that's just being transported in. Overall, we felt that it was pretty decent coverage, but you have to be pretty careful to understand that the DNA does not mean that the actual species are present. If we look at that a little bit more closely, these are 3 of the sites that were sampled in Great Bay. So Nanny's Island and Adams Point are kind of out in the bay. The Lamprey River site here is actually very close to the Lamprey River. At low tide, it's practically sitting in a mud bank. If you look at the number of samples collected and the species identified, for the 2 sites in the estuary, it's fairly similar. We're seeing an increase of samples, and this is, I think these samples are all collected in June. Increase in samples with increase in species with more samples. At the freshwater site, we see, or the site with freshwater input, we see significantly more species, and that's because we're seeing or detecting those freshwater species, or at least we're detecting the DNA from those freshwater species, which is not the same as detecting the actual species. And then we can also look at the species, and we can look at sequence variants. So this is, again, number of samples, and this is sequences which map to a particular species with a high degree of confidence, but which are not exactly the same. So there may be one slight difference, which indicates that at the very least, there are a different number of individual fish present within that species. We did look at these accumulation curves for other sites, and they tend to be similar. For most of these sites that we worked with, we don't have as many samples. B, some of them were taken to look at different questions, such as whether or not anadromous fish are returning. So we don't have a estuarine sample set for each of these, but if we look at them generally, we see that the curves are similar. This is Apalachicola in Florida. These 3 are the Great Bay curves that we already looked at. In Apalachicola, we see slightly more species, and that's probably because it's a nice warm environment and they just have more species there. But again, for any of these sites, or for none of these sites, if you put it that way, we do not see this curve leveling off. So as you continue to sample, as you continue to look at more sites, or you add more time points, you will continue to see more species. So what are those species that we are seeing sort of at the top of the curve? What are those things that we're detecting as we increase our sampling density? What we found, this is Great Bay, 2019 again. The most common species are detected multiple times. So there are about 19 species that we detected at least 5 times. And those are species that we know are present, that we expect to see, so they're common, and we commonly see them in the DNA. Then there were 17 species that were only detected once. And those are very likely to be species that, like haddock or rock gunnel, that are probably present in the area, or that may be present just offshore. So it's possible that the DNA comes into the estuary, but they're not very prevalent. So it's not entirely surprising we don't see them. But if we wanted to do a really thorough survey of the estuary, again, we'd probably have to have more samples in order to pick those up. The thing that's perhaps most interesting are these species, like we got northern kingfish detected in 1 sample. That's not generally present in our region, but it's south of us. So as we think about using something like eDNA for looking at invasive species, for looking at range changes, we want to be really careful looking at these very rare detections. So this could indicate that there is some DNA from a northern kingfish, whether it's actually there or whether it was carried up from somewhere in the vicinity. It could be an error. It could be something that got transported somehow. And the only way to really find out is to keep sampling. So you want to take multiple samples. You want to continue to see if that species turns up, and in which case that will give you an indication that it is actually present in the system. And one of the things that's really, really important we found when we're working with these eDNA detections is that managers need to understand what a detection means. If you tell them we got northern kingfish DNA or we got rainbow trout, and they believe, they know that that species is not actually living in their estuary, you're going to erode trust. It's problematic for describing the method. So we need to understand that DNA does not mean that the species is actually there. It probably means that the DNA is there. So you need to understand what transport mechanisms could have brought that DNA there, or if there's a possibility that that species is actually present or possibly ephemeral, passing through and then moving on. So interpretation, the sampling needs to be carried out very carefully. The analysis and interpretation is the second step, to which we need to pay at least as much attention as we pay to the sampling and analysis. And I think that's a step that often is not perhaps gives as much attention as it needs to. So overall, we found that eDNA's monitoring can be really effective for surveying fish communities. Remember, again, these are samples that are taken as part of an existing program. So you don't have to go out there in a separate boat. It does take a little bit more time. The filtering takes a little bit more time, but it's not a huge add-on to an existing program, and you get a really different set of information. Again, emphasizing that the presence of DNA needs to be interpreted very carefully. Doesn't necessarily mean that organism is living in that environment. We also found that it's particularly useful at locations where net sampling is not allowed. So if you want to do a fish survey in a place where there are endangered species, where it's a sensitive habitat, you can use the DNA sampling to get a survey of fish species without having to go in and do any of the damage or the stress that might be involved in a net sampling. And again, just emphasizing, it's a great way to leverage an existing water quality program. So if you're out there taking samples anyways, particularly if you're taking nutrient samples and you're filtering, you already have the setup. It's pretty easy to do. So overall, we were really encouraged by this. We feel that it's potentially a great method to expand existing survey capacity, and we really hope that other people are interested and that we will start to see this really great potential realized in more areas. We do have a website where we talk about some of these results, and if you have any questions or any thoughts or comments, do please reach out to me at AllisonWatts@unh.edu and let me know what you think. Thank you very much.